Our next speaker is Dr. John Galgiani, who will present challenges of clinical trial designs for therapeutics and vaccines. And John joins us from University of Arizona College of Medicine, and he's also the chair of the Valley Fever Center for Excellence. John.
Thank you. Hi. Let's see. Which way is it? This way? Oh, good. So I'm John Galgiani. I'm from the Valley Fever Center for Excellence at the University of Arizona, and I'm going to be talking about strategies. And I have a couple of disclosures. One is that the University of Arizona is continuing to be the sponsor of a drug, Nicomycin Z, with the FDA, and I'm the signature on that IND. But I used to be affiliated with Valley Fever Solutions, which has the license for that, and I'm no longer doing that. And also, as you'll hear later this afternoon, members of the Valley Fever Center for Excellence have discovered a vaccine, and they'll be presenting that work as a vaccine currently in development for dogs, and that's very interesting stuff as well. I didn't need to put on this comment. The opinions expressed in this presentation are my own, but I put it on voluntarily because I wanted to just sort of emphasize that some of the things I say are meant to actually stimulate discussion and may not be completely shared by everybody. Some of what I'm going to talk about I know overlaps with what John Rex talked about yesterday, but I think we'll probably put slightly different emphasis on it. So let's go a little back in history and mention myconazole, which George Thompson put on his slide in the 70s. Hilly Levine had actually access and was actively working on cocci in mice at Oakland Naval Biosciences, and he was getting drugs to test for dermatophytes activity there, but because he had the mouse model for cocci up, he started testing some of them, and one of the drugs he tested was myconazole, and had some spectacular survival differences between controls and those treated with myconazole. David Stevens at that point was transitioning from his previous research career in virology into mycology and saw the results that Hilly showed him, and almost immediately found some myconazole to give as compassionate use for one patient, and then another, and then another, and then another, and then another. And then after a while, the FDA said, you really need a protocol for this. And that led to really accumulating quite a large sample of pretty much David's drive, which resulted in the drug actually becoming approved by the FDA, although it's as a parenteral, not as a spray for your feet, but as a parenteral drug. And his presentation of his data in published reports was either response, partial response, or non-response progression. And that was in debate as those publications came out as to what do you mean by these things. Bill Dispukes, the next decade, published an article in which he, among other things he did, was publish criteria for evaluation of therapeutic response to antifungal drugs. And he proposed criteria which enabled quantitative comparison of patient status before, during, and after therapy. And we tried to implement that in a randomized trial of 400 milligrams versus 800 milligrams. It's 400 with or progression to 800 milligrams in cocci. And the way we used a scoring system at that time was to treat everyone as no response with a 0 response at the time of enrollment, and then adding points with improvement. And the problem with that, it worked, and we published the data, but what you ended up with, some people with relatively minor spectrum of disease and others with massive spectrum of disease. So the actual scores really were very different across the entire population that we were studying. That was refined and then used in future studies in the concept of flipping that, so that at the time of enrollment, the patient had 100% abnormal, whatever that was. And then as the patient improved, reductions in the scores were evidence of improvement. And then because we didn't want to use this as a continuous variable, we wanted to score patients either as responders or non-responders, because patients are not a continuous variable. They're not digital. They're I mean, they are digital. They're one or another. So they're either responding or not responding. You had to affix some criterion of response, such as 50% improvement. And let me just emphasize that if you do that, there are some very important little details that emerge as we did that. First of all, you have to, everything that you set on the baseline, you have to repeat. You can't start changing around the things you're looking at, because otherwise things don't work very well. And a correlate of that is many things didn't get repeated, like imaging studies, for instance, that were expensive and weren't being paid for by the studies. So anything that you didn't evaluate on a follow-up evaluation needed to not change the score. You can't just assume it's changed. And that was a very conservative decision, but it's very easy to see how important that was. And then following, the other thing is that we realized that really you never got follow-up cultures on almost anybody, because most of those cultures required invasive procedures, and if the patient got better, no one was very interested in just a biopsy, again, just to see what it would show. And so within the NIH-funded mycosis study group, we had multiple studies these were almost all phase 2s. There was one phase 3 with pretty substantial groups of patients. And that was over a period of some 8 years or so. And I'll focus on that one phase 3, the randomized trial of fluconazole versus ITRA, which had nearly 200 patients randomized almost evenly between the two groups. And this brings out another point in clinical trial design. Here you can see, using the scoring system in terms of response, that ITRA goes better than flu, but we had to have a primary hypothesis. And we define the primary hypothesis as 8 months. And so the difference between response rates of flu and ITRA at 8 months was 0.08. Subsequently, at 12 months, it was actually significantly different, but that was a secondary analysis. So technically, the study failed to show the difference because you rise or fall on whatever you decide to be your primary hypothesis. And that's actually sort of a critical issue. You can have a very good drug, but if you design your hypothesis incorrectly, you may miss the observation. So to summarize the MSG scoring systems, it is a quantitative description of the proportion of clinical response following initiation of therapy. Patients with many different manifestations actually fall on the same scale, by the way we implemented that. And I think, generally speaking, Royce is here and others who were part of those studies sort of thought that the response as scored seemed to look like what the patient looked like. So we were all kind of happy. We weren't scratching our heads why the score went one way and the patient seemed to be going another. The cons are this has never been validated, and John Rex emphasized that yesterday. And so without validation as to what this score is representing, it's going to be a tough sell, I think, at the FDA. The exact criteria and methodology for scoring are not agreed upon. There's lots of interest in revising it, and that's fine. But even with revision, again, it still would have to be validated, whatever that is. And I'd like to emphasize that since these measurements need to be repeated exactly as baseline, it's kind of difficult to try to validate the scoring system on retrospective data. You really need to collect your data prospectively to do that. At least that's my sense of how you'd have to validate it. So I'd like to also point to actually the way we are getting drugs to treat patients right now. All drugs, as was pointed out earlier, are off-label. So we have miconazole is not in use. Ketoconazole is essentially not in use either. Amphotericin has its problems. So the drugs we are all using, oral azoles primarily, are not have the FDA indication for COXI. And when we had the NIH-sponsored MSG, we still had published data with large sample sizes, or at least relatively large sample sizes, so we could make determinations as to what the error bar was about response rates, at least by this MSG scoring. But since then, such as drugs such as posaconazole or isovuconazole, there have not been large, in fact, there's surprisingly little information in the literature, for example, for isovuconazole, despite the fact it's now increasingly in use. So one, I think, strategy is to, and John mentioned, if you have the chance to develop your drug for a different fungal infection other than cocci, do it. And that actually has been the route, so that drugs get approved for something else. They become available. They're used off-label by clinicians because they need to do something for their patients. And I actually pulled out this data. Actually, David Nix, one of our clinical pharmacists at the U of Arizona, pulled out this data from Medicare. 2020 was the data most available. And I showed Arizona on the top line, and then Four Corners and Nevada on the following four lines, and I lumped Colorado and New Mexico together as not having very much cocci, but their population over 65 is about the same as Arizona. And the point I'm trying to make here is you can look across Arizona at how much drug and money spent on that drug is being used, where cocci is very prevalent, and in contrast to that, how little of posaconazole or isovuconazole is being used in Colorado and New Mexico combined. So there is profit being made on treating patients with cocci by drugs that do not yet have FDA indication. So the challenges, I think, to summarize, to doing randomized controlled trials for cocci is the need to have an FDA-approved endpoint. The scoring system has its problems I've just mentioned. Would patient reporting outcomes be the right answer? I think that's a really interesting approach, and it's nice to see John Rex trying to develop that. I actually was at a vaccines meeting last week and heard of a patient-reported outcomes measure that's being used for RSV, and I think so there's many other respiratory patient-reported outcome things that could be used. Then you need an appropriate companion drug. Since there's no approved FDA drug that would be appropriate for this study, you have to figure out which one you want to do and why to the FDA's satisfaction. And since there's no approved drug, you really can't do a non-inferiority thing because no one's shown efficacy for an approved drug. So let me also touch on the challenges for doing randomized controlled trials in primary cocci. And I noticed that John Rex put a little X across this possibility, and there are real challenges with that. It's never been done before, and there have been several starts. Only feasible endpoint is reduction in length of illness. You really couldn't get sample size to look for complications because they're so infrequent if you start with your denominator being people who are being initially diagnosed with early pneumonia. And then, in fact, many of the symptoms that are generated by primary cocci, and I think Neil alluded to this, have very little to do with actually the fungus proliferating. They have to do with the immune response that's generated, and whether or not treatment of the fungus with an antifungal will have any bearing on those symptoms would be confounding, I think, in terms of interpreting drug effects. And then the other issue is no matter how you design the study, you may be accused of not getting the patients enrolled early enough, and that's why you miss seeing the effect of the drug. There are strategies to try to avoid that, but those strategies also have the problem of increasing the sample size maybe fivefold over what you do otherwise. So let me turn second to the problem of developing vaccines. And I just mentioned that I think there's ample validation for why a vaccine is justified in terms of, even though it's a small market, it clearly has public health and economic impact. And as Reba talked and others have talked about, if you could make a diagnosis of fungal infection, you stop using antibacterial drugs empirically. And this was the survival curves of data back in 1965 showing you mice not vaccinated in the controls or vaccinated 1 time, 2 times, or 3 times with a formalin-killed spherular vaccine that was 0.8 milligrams per dose per mouse. But we were very struck by that efficacy in those studies in mice. And that actually led to a clinical trial of the FKS vaccine, which I was a part of in the 80s. It was also done in Bakersfield, Lemoore Naval. We skin tested those people 4 times, 2 usual strength and 2 high test strength, and only those that were negative of all 4 vaccines were included. And in doing that, we excluded a chunk of people who wanted to volunteer for this. And the serologies were then followed periodically. We were able to get 3,000 or so, 3,200 person years in the study. The first thing to point out is how intolerant or how difficult and how inflammatory this vaccine was. And this alone probably makes that vaccine not something to go forward. And it's not surprising. If you look at the dose these people were getting, it's 1.4 milligrams per dose times 3, and shown there is definite infections. There were 9 in the formalin-killed spherular vaccine group and 12 in the placebo, which is not a significant difference. But this showed, first of all, how far we've come, I think, for the cocci people to think about what would be a useful vaccine. This vaccine would never even be considered now as a potential clinical trials vaccine. And second of all, I think it also shows that you can do this study. And I just quote a few sort of what ifs here. If you had 15 cases in your placebo, shown here is if you had 4, 3, 2, or 1 cases in your vaccinated group, you actually see significant differences with fairly good confidence level. So I think you could actually do this study, and I think Tom Manath later this afternoon may talk a little bit about trial design. So the challenges for doing randomized controlled studies in vaccines is what population should you study? You have to make that decision. Should you exclude people with past? I think that's an issue, whether or not the FDA would allow you to do that exclusion. What bias would you introduce into the study? Endpoints are being discussed here, and I think any symptomatic infections would probably be the endpoint, but all of those kinds of details would have to be worked out. But I think the study we did do with the FKS vaccine shows that it probably would be possible to do this study if such a study was embarked upon. Thank you very much.
Thanks so much, John.